Okay, great. Can everyone hear me? Yeah. So it's a pleasure to be here. I'm in the systems biology department at Columbia University at the medical center, and this is my kind of first interaction with kind of people in this area. But I'm very excited about kind of the program here. And we're in the systems and synthetic biology area, and so we're primarily experimental group. But today I wanted to share with you a story that we have been working on for the past few years on trying to explore the possibility of genetic information compression, and then I'll tell you what that actually means, and I'll try to keep us on time. So it turns out that from the first genome that was sequenced by Fred Sanger back in 1977, in this famous paper now of the Phi-X 174 genome, which is a bacteriophage, it's a single-stranded DNA bacteriophage, they found something very peculiar, which is that two pairs of genes are actually encoded by the same region of the DNA in different frames. And this kind of actually pushes the boundaries of what we think about in terms of information encoding within DNA, that we typically think that essentially one piece of DNA encodes one protein coding sequence. But it was very evident from the first get-go of having done full genome sequencing that we're starting to see these kind of peculiar kind of information encoding regimes. And what this really means is that, so this is just a representation of the Phi-X genome here. And so that you see that sometimes some of these genes are actually encoded in different frames with other genes. And so this actually, it turns out to be a very common occurrence rather than a rarity. And so this kind of brings up this idea of natural overlapping genes, and kind of, it turns out these are actually really, really prevalent. And so I don't have to kind of point out that essentially you have on one frame here, you have the opportunity to start in one frame to generate a protein. But because of the kind of the triplet codon nature, you could potentially have a frame shift into a different frame and generate a totally different protein if you initiate translation in a different frame. And certainly that also is true for the +1 frame, +2 frame, as well as the reverse frames. And so in principle, it's possible to get multiple proteins from the same DNA sequence. It turns out that about 30% of bacterial genes have these overlap, at least partial overlaps, if not full overlaps. And there are many examples of this also in eukaryotic genomes. And this is actually very frequently found in viruses. It's actually quite amazing, this example here, where this is the HBV virus. It infects about 350 million people worldwide a year. And it's a simple 3.3 kb virus. But within this virus here, especially one of these genes is a polymerase gene, which takes up almost all of the entire phage, or the virus. But within this virus, you have essentially a surface protein completely embedded in that gene. And so this idea of kind of information compression is really prevalent. And so kind of what's the benefit of why do biology do this? So, of course, from first principle, you might think that this is potentially a way to layer additional units of information on top of one another, such that you can kind of decrease the risks or the catastrophic events of in the face of high mutation rates. So viruses tend to have much higher mutation rates, for example, than kind of free-living organisms, because they have to constantly evade the immune system. And so if you, for example, generate a synonymous mutation in one frame, that could actually lead to a deleterious mutation in another, and kind of vice versa. So you essentially have these coupling of synonymous mutations in one frame with non-synonymous mutations, and it turns out that that actually can improve kind of the fidelity, or at least direct the rate of mutations in a way that enables both kind of keeping the information as compact as possible, but also kind of moving the evolutionary kind of along the evolutionary landscape in this other way. And so there's lots of kind of studies on this. I won't kind of go through all the literature. And so as an experimentalist and one in synthetic biology, we were very kind of interested in this because we see this in nature, but we've never been able to generate something of this sort. And so we asked this very simple question, which is, could we actually generate these overlapping genes de novo? And first of all, is it feasible for us to do this? There are lots of existing natural overlapping sequences suggesting that the protein space is like quite rich, that you might functionally get the same protein function, but have many different ways to encode that. And so that's, from the get-go, what is the real information density of a functional protein, right? And the second question is, can we actually do this? And are there enough data sets and models and computational tools for us to try to achieve this de novo design and actually try to experimentally test it? And then, what are possible uses of this for kind of a forward engineering perspective in terms of keeping evolutionary stability or biocontainment? And so we came up with this algorithm, which we call constrained adaptive mutations using engineered overlapping sequences, and I'll go through what that is. Essentially, we're trying to take two sequences and trying to smash them together. We have the ability to encode on the synonymous space, but also we have to make non-synonymous mutations because there's just not enough flexibility in the code for us to just make synonymous changes that allow for kind of two sequences to kind of converge together. And so we have an algorithm that kind of breaks it up into two pieces here and ways to improve that. And this is something that we published earlier last year. And so kind of the first question is, are there good representations of protein and protein function that we can use? So it was mentioned that there are different kind of representations of this. So protein hidden markup models are really kind of interesting ones. You basically take a multi-sequence alignment. You can generate these kind of position-specific, based off of residue conservation, position-specific insertion, deletion, and mutation probabilities, and then you essentially kind of take the minimal kind of probabilities to the crossings, and that gives you some representation of protein based off of what evolution has experienced that we can kind of learn from. So when we actually try to build proteins using these HMM models, it turns out that they're actually not functional. And one of the key reasons for that is that we're missing these long-range interactions. And so, if you just take the average protein, it actually doesn't tell you anything, right? And so it turns out these long-range actions are quite important. And so that's summarized basically by these, instead of just having these nodes, you also have these edges where specific pairs of residues are essentially co-conserved through some evolutionary process, usually kind of associated with kind of some type of interaction from one part of the protein to another that helps with folding or some other functional processes. And so there's good algorithms to do that and to generate these models. And so we use kind of essentially these two things to try to help us to guide kind of this de novo design principle. And so I'll just go through how we algorithmically think about this very quickly. So this problem essentially can be distilled down to overlapping into tetranucleotides, right? So each tetranucleotide can encode for two residues. And if you have these tetranucleotides overlap with one another, you can generate this kind of this scheme here where you have the conservation of position 1 and 4, and then whatever you're optimizing in the middle here. And so the idea is you're optimizing, balancing between the best bases for the current and the subsequent positions and using this. And so this allows you to essentially get that frame that you otherwise don't get by simple encoding. And so, so you essentially take these tetranucleotides based off of the HMM representation, and then you find essentially the maximal scores associated with those by essentially putting down the position 1 and position 4, and then you can calculate everything in the middle. And the nice thing here is that then you can essentially chain this and having multiple of these tetranucleotides across, and that gets you to basically, you could extend this and tracking essentially a number of sequences down this chain in a very tractable way. And so this is just represented here where you essentially take the optimal path to these tetranucleotide sequences, and that gets you kind of the best scores based off of the HMM. And then you can, of course, seed this by doing some random kind of mutations and suboptimal paths that gives you kind of even more seeding scores. And then the kind of the kicker here is that that was just to get the seed, and then we then go through using these long-range interactions to then optimize further to get kind of these long-range kind of co-associations by iteratively optimizing from these pseudo-likelihood scores. But so I want to say like this is actually work, right? So computationally, this is how this is we can do it. Turns out that we can't get this to work, otherwise I wouldn't be telling you this. And so this is one example of this. And so this is taking threonine dehydratase, which makes an essential amino acid, isoleucine, and then this acyl carrier protein, which is an essential gene that's actually the most abundant gene in the protein. And so we can computationally essentially take the wild-type protein sequence and redesign the ILVA protein into this region of the protein here. And so we can put this in a cell, and the cells are able to grow when we remove this essential gene from the genome, and then both of these genes are functional. So the question here is that, now that we have a cell that relies on both of these co-coupling, what are the evolutionary consequences of this? You can do high-throughput mutagenesis of this, which we did to experimentally test if you make a mutation in these overlapping regions, how does that affect the cell? So if there's an extra copy of this gene, so in this case, this gene is an essential gene, so if the cell doesn't have this gene, then it will not survive. If you have an extra copy of it on the genome, any mutation in this region has no effect, right? So this is basically all 64 codons. There's no effect. But if you essentially remove the wild-type copy that's in the cell and essentially relying on this overlapping region, what you see are really interesting patterns now. So these correspond to essentially synonymous mutations that otherwise would have no effect. Now they have significant effects. So this is an example of this is a leucine codon, for example, at this position 4 here, where 4 out of the 6 encoding for otherwise synonymous leucines yields very deleterious mutations. So you can see that based off of the fitness map here. And that essentially you could explain some of this very simply by showing that essentially this creates a stop codon in the other frame. And that's why these codons are not good compared to these other ones. And you could really start to look at, kind of explore the variance of this and kind of the robustness and degeneracy of the code in this way. And so we can kind of do this in high throughput. We don't want to test this one at a time, but we could explore many, many different versions of this. And so we kind of developed a way to experimentally test thousands of these combinations at the same time. So about 7,500 encodings of these pairs. So this is another example of taking essentially a cysteine biosynthesis gene and smashing it with an initiation factor gene. And these are kind of just possible ways that you could get this. And so kind of the key surprises here really is that some of these you can get very low homology, so wild type, so something like 26% protein homology. And it turns out that one of these proteins is much more kind of conserved than the other one, which kind of gives you some idea of potential optimization along this kind of on this landscape here. And there's lots of these epistasis that kind of we seem to observe here. So kind of just to conclude here, I just want to at least have the takeaway that these current statistical methods, I think, are sufficiently powerful enough that we can start to do computational designs. These sequence entanglements, as we call them, help to stabilize DNA against evolutionary drift. And you might be able to, for example, encode other things like toxins to prevent it from escaping. And maybe there are some other opportunities for this for data encryption and things of that sort. So this work was led by a really talented graduate student and a postdoc in the lab here, as well as just thank my funding sources. Thank you for your attention.
Time for one question while Eric comes up. Go ahead.
Thank you.
We need a microphone here.
Thanks. That was really interesting.
I just wonder if when you look at the coding for that second, the overlapping gene to be expressed, it also needs a good ribosome binding site, and stuff. Do you include things like Shannon-Dalgarno sequences and stuff, or just purely the coding region itself?
Yeah, so that's a great question. Yeah, we add we do some synonymous mutations upstream of the embedded gene, so to kind of make a better Shannon-Dalgarno sequence for translation. Yeah, it's actually the harder part, it turns out, because tuning the ratios of these is not quite trivial.
One.
So I just wanted to give you one remark and ask one question, and I'll do it really quickly. So what you're showing us is really an algorithm that coding theorists have developed in the past, Viterbi's algorithm, which is really a pride and joy of the information theory community. The other thing, that was just a remark. And the other question that I have is basically when you run Viterbi's algorithm and you keep mentioning you're optimizing the selection process, what is the function that you're optimizing over? What is the objective function? And second, why just stop at 4 tuples? Why the complexity is not that high? I guess you could go a little bit further, maybe merge 2 codons, because there is definitely dependence between the amino acids that are stitched together in adjacent order.
Yeah, so the first question here is, the objective is basically the pseudo likelihood score that we find associated with its likelihood to have coupling, right? And so this is both integrating both the edge energy and the node energies. And so this is based on multi-sequence alignment, so certainly there's other ways to do that calculation. You could do evolutionary coupling and other things. And so the underlying question is, what makes a functional protein, and are there what is the information content of that? There's many ways to make the same hydrolase, right? And so, but we don't actually have that good representation, and so this is kind of the underlying question. And maybe the second question I could take.
You're looking at the codon, the
triple,
and then an innovation symbol, so you could go probably higher if you have enough computational power, and that would probably give you better constructs in the end.
Yeah, I think at the end here, the question is really about, like, we could design as many things as we want, but testing it is really ultimately the limitation. Lots of things work by computation, they give you the same scores, but that's not true in actual experimentation. So.
Okay, well, thank you very much.